Hello, today we'll be talking about lab safety and proper lab protocol. So before you enter the lab, you want to make sure you have everything with you. That includes having your PPE, so that means glasses, lab coat, gloves. Make sure your glasses are on you at all times. Then you also want to make sure that you have access to the lab so that you're authorized to do work. So whether you're a TA or you have the card to enter, or you're doing it in the proper disposed lab times. Usually there'll be a sheet on the door when you're approved. Next, we want to talk about proper safety precautions. So knowing where the eye wash station is, which is right there in the hallway. If you have any problems, you go over there. As well as where the shower is. So going straight off to the top, we have our shower here, along with a fire extinguisher. This will be checked every month. And if you have any need to use it, it's over here. Moving forward, this is what you'll see from the lab as you enter from this entrance. And we're going to go walk around to see what we're going to see. Make sure, importantly, to follow the instructions of your supervisors or your GAs whenever you're in the lab, as they will ensure your safety and the safety of others as well. You need to make sure you know where the proper waste is. So to your right will be the biohazard waste. So anything biohazardous, anything contaminated, anything that could cause danger to others, you want to dump in here. Gloves that are not contaminated should not be dumped in here, neither should paper towels. To your left is the sink. Make sure you wash your hands before you start doing work. And this is what one of the stations looks like. In here, you'll see a crock with an orange bag. This is the biohazard bag, and this is where you dump any biohazard waste here, and you finally dump it into the big container as shown previously. Any tips, any contaminated products, any spilled chemicals should be dumped in here, and here only. Make sure you properly wipe and disinfect the surfaces. As we go around, this is where the first aid kit will be. If you need it, this is where it will be. Make sure you notify your GA or TA before you use it and of any injuries in the lab as soon as possible. Next, you'll see another fire extinguisher to your right. As we walk around here, you'll see another station with tissues as well as the ethanol. So before you start, make sure you wipe everything down in order to ensure no contamination to you or your teammates. And make sure the surface is clean so no contamination for the experiment as well. As a reminder, make sure paper towels are not disposed into there and to dispose them into a normal trash can, so we'll carry it with us as we go. You'll see here sinks on either sides as well. Make sure you wash your hands before and after you use the lab. So before you enter and before you leave, make sure you wash your hands. Along here, that's a station with another sink. This is where you'll clean your glassware. Another fire extinguisher along with a water bath. Make sure you know where those are. To your right is the SDS pages. And then as we walk through the lab, we come to the entrance where the main trash can is. Any waste should be disposed into there that isn't contaminated, as well as any gloves. And that's it for the introduction. We'll go over SDS pages. This is the SDS pages, the safety data sheets for both the materials as well as SOPs or standard operating procedures for emergency responses. You'll see here both will be located towards the back exit, and within each one contains all the lab equipment and all the lab materials you'll be using, as well as the proper precautions. In this case, that's your kits. And you can see here that the safety data sheet for everything is included here. If you have any questions, ask your GA or TA, but also feel free to consult them here as well. Every reagent, every chemical, every protocol is here. So make sure you consult them if you have any questions. So this is the safety data sheet page, and it's updated every time, every semester. SOPs for emergency responses is what will be done in case there's an emergency situation. So whether it be spills, cleans, fires, either they have to evacuate, who to contact, what to follow. Normally your GA or TA will take care of this, but in case you want to refresh your memories, feel free to consult them at any time. As well as if you have any questions, feel free to ask either the GA or TA or your lab coordinator on the proper protocols and the proper techniques. And that's about it. So first we'll be talking about the different equipment that you'll see in the lab. So before you start anything, you're going to want to approach and wash your hands and make sure you dry them off. Then you'll see that at every station you'll have the similar setup. So before you start, you want to first grab your 70% ethanol, and you're going to want to spray down and wipe your surfaces. Now that your surface is clean, we'll be going over everything that you see. These are your pipe headers. They'll come on a rack and you'll have the different sizes. The sizes will be written on the side like shown. You have different sizes ranging from 1000 all the way down to your 2 microliter pipe header. And the way you use it is you push down all the way to the first stop when taking up, and then when you're about to dispense, push all the way down to the second stop and make sure everything is gone. Now you'll see that each pipe header has its own tip, and here's the tip box. The tip size will be written on the top, so you're going to want to use that to equip, and then anything that you use is thrown onto this, the biohazard disposal. So throw out all your contaminated tips, and finally there's water if you need it in case the experiment asks for distilled water. And also you have your racks on the side. All stations will look very similar, so make sure everything is there when you need it, and make sure when you're done, everything gets returned to how it was. Now, walking around, you'll see a very similar station on the other side. You'll then see a microcentrifuge, so if you have something with small levels of G's, as well as microtubes, you put them into that microcentrifuge. More information will be regarded when discussing it in the proper video. Next, you have your power supply, as well as its reservoir. This is used for DNA and Western blotting. This is the tank that's used for Western blotting. And in the appropriate video, we'll go over a bit more, but this is what it's looked like just to familiarize yourself with them. Next, we have our heating block. So we want to boil something, especially in Eppendorf tubes. We use this, mostly used for Western blotting, but it might be used for other purposes. Please follow the proper video on knowing how to use it and the proper safety discussion. Another sink on the side of the class. Here is a vortex. It will be used very often when you need to mix your reagents, make sure they're homogenized. It will be used mostly, and the way you do it is you turn it on, and it will vibrate, so you just put your tube, or you put your Eppendorf tube on the top, and it will mix it, and you can adjust the speed like I just showed. Next, we're going to go over to the side of the station, where you have your TAE buffer, you mostly use for DNA blotting. Then you have your scale, and you also have a shaker. Depending on what you need, you'll use it as needed. So it's on this side of the class. Make sure you familiarize yourself with the situation, and make sure you accurately weigh yourself. Additionally, a pH meter. When you make your buffers, you want to make sure the pH is correct, and please use the following video appropriately to know how to use the pH meter, how to calibrate it, as well as how to judge the pH of your buffers that you make for your gels, as well as your Western blots. Next is the microwave, used for heating up elements in your buffers for the DNA electrophoresis. Graduated cylinders, along with a wash station, in case you need to wash any glassware. And finally, distilled water, in case you need to refill up your water bottles. So just familiarize yourself with this station. It's not too important as your main station, but you will come here from time to time depending on the experiment. Next, moving on, is the SDS books on the right, and then the thermal cycler. We made a whole video about it, so watch that, but you'll use it during DNA PCR. The proper video will be shown how to properly use the software itself, but make sure it's clean and as shown. Next is your shaker, so you'll probably leave your membranes here when blotting in primary and secondary antibodies. A proper video will show you how to use them and how to set them to the correct speed, but they'll be located on this side of the classroom. Here's the second one. Next is your microplate reader. It will look something like this, where you'll read your microplates, as well as the ChemiDoc, which will be shown for your gels. Both your Western blots and your gel electrophoresis will be imaged through this machine. And finally, your macrocentrifuge, used for reaching the higher levels of G's, which is impossible in the smaller microcentrifuge. Please handle with appropriate care and make sure everything is balanced. A proper video will be shown how to use that. Next, you have your fridges. These are mostly going to be used by the GA, but if you need to put your things in there, they'll be as instructed by the GA himself. You have incubator shakers and incubators. These will be used by the GAs, so do not touch them unless instructed to. And that concludes the lab tour of all prominent equipment that you'll be using. And now the last thing we want to talk about is the fume hood. Some experiments will require you to use the fume hood because the chemicals can be toxic and the fumes they emit can be harmful to you and those around you. So you're going to want to open it from the bottom here. So you'll see that this is the required height. So you want to raise it to that height. You're going to grab from the bottom and you're just going to want to simply pull up. And you want to direct it straight to the height. This is the height that will ensure that the maximum airflow is achieved and you are protected the most. And then you're ready to go. So you can just go in, do the work you want to do, and all the airs and fumes will be sucked up. Just a quick tour of the other things on the side. You have cold water here, you have vacuum, and you have an air. And finally, gas, depending if gas is loaded in. You probably will not be using these, but they're there in case you need them. And this is how you set up. And when you're done, I'm going to grab it and just push it all the way down. Pushing it down might be a bit difficult, so make sure you go all the way down until you can't hear a sound anymore. It's not properly closed. Slam it all the way down, make sure it's making full contact at the bottom, just like that. And now it's perfectly closed and ready to go. And that's how you go over the fume hood and how you'll be using it in your labs.